From our studios in New York, this is a special edition of Charlie Rose.
Tonight, we continue our exploration of what many consider the most fascinating topic in science, the human brain. In previous episodes, we examined how the brain controls vision, movement, and social interaction. Tonight, we will look at how the brain actually acquires its amazing abilities. Our subject is the developing brain. Every newborn is introduced to a world that is entirely foreign and unknown. However, the adaptive ability of the infant brain is nothing short of miraculous. Within days of birth, a newborn starts building the foundation for a lifetime of knowledge. He or she learns to respond to colors, to recognize objects, and to discern the properties of the physical world. In less than a year, most babies have already begun understanding language. Tonight, we'll look at how this learning takes place. What cognitive tools do infants use to understand the world around them? How do they acquire language? Are these skills learned spontaneously, or do they require outside instruction? And perhaps most importantly, what kinds of abilities are innate, and what kinds of abilities must children get on their own? Findings show that newborns possess much greater cognitive capacity than previously thought. Skills such as object recognition, facial recognition, and even basic number recognition can be inborn. One thing is certain, babies are much more than empty vessels waiting to be filled with knowledge. They are primed from birth to gather information on their own. This evening, we'll also learn about the anatomical changes that occur in the brain as a person grows from a fetus to an adult. Just like every other part of the human body, the brain begins as a single sheet of cells. Unfortunately, aspects of the growth process occasionally go awry. Tonight, we'll also focus on a handful of diseases known as the developmental disorders. Rett syndrome, Fragile X syndrome, and Fox-PT syndrome all arise from defects in single genes. Each interrupts a different stage of brain development with devastating consequences. Joining me tonight, a remarkable group of scientists who have devoted their careers to understanding the developing brain. They are Elizabeth Spelke. Her work has shown that infants are born with much greater cognitive abilities than ever before imagined. She is a cognitive psychologist at Harvard University, where she is director of the Laboratory for Developmental Studies. Patricia Kuhl. She is one of the world's leading authorities on speech development. She is a professor at the University of Washington, where she is director of the Center for Brain and Learning Sciences. She is also the author of The Scientist in the Crib. Huda Zogby. She studies the genetic origin of developmental disorders such as Rett syndrome. She is director of the Jan and Dan Duncan Neurological Research Institute and also a Howard Hughes medical investigator. Stephen Warren. His work focuses on understanding the causes of mental retardation. He is a professor at Emory University in Atlanta. And once again, my co-host is Dr. Eric Kandel. As you know, he is a Nobel laureate, a professor at Columbia University, and also a Howard Hughes medical investigator. He has been our companion, he has been our guide, he has been our guy on understanding the brain. Good to see you again.
Pleasure to be here.
So we begin with the question, what do we mean by the developing brain?
We are exploring together one of the great miracles, as you indicated, in the natural world, the emergence of the human mind. This is an extraordinary adventure. An infant comes into the world with an enormous curiosity to learn and acquires these abilities systematically. Not only do they learn very specific things, to recognize faces, to recognize objects, but they learn abstract things. They learn how to acquire language, they acquire numerical systems. And what is interesting about this is that they don't do this instantaneously. They don't acquire all of these skills simultaneously. They do this in stages.
That becomes this name, who is famous for understanding the developmental stages.
Jean Piaget.
The origins of intelligence in children.
Absolutely. This man is a giant in developmental biology and understanding the steps whereby kids learn about the natural world. And he came across this in a very interesting way. He found that it isn't just a question of kids getting more intelligent with time, becoming more like adults in terms of acquiring information, but found that their qualitative thinking changes. They are capable of handling certain kinds of information only at certain stages in development. So that at early stages they cannot understand certain processes that are very obvious to us. And the way he came across this is he did most of his work in Switzerland, but he spent some time in France, and there he translated the IQ test into French, and he gave this test to lots of young kids, very bright kids, and he saw that at certain ages, invariably they got certain answers wrong that seemed very obvious to him, and that a few years later they would be able to do very well. Let me give you an example. So you have two small beakers with an equal amount of fluid. If you take one of these beakers and pour it into the larger one, you will see that you now empty this one beaker and you fill the other. Let me try this as an experiment with you, Charlie, to see whether or not you can manage to do this. Two blue beakers. If we now pour this into that, and I ask you a very simple question. Which beaker has more fluid? This or that?
They're the same.
Brilliant. You pass. Kids don't realize that the volume stays the same irrespective of the shape of the vessel.
Because
they think if it's tall, they think it must be bigger.
Yeah.
And it's sometime 2 or 3 years later, when they're 7 years old, they all of a sudden, boom, get this perfectly well.
You know what amazes me, Eric? I mean, here's one more, Jean Piaget. I think born like in 1889, somewhere right before the turn of the century.
That's right.
Here we are talking about one aspect of science that is right at the cutting edge, where there's so much unknown, but we're learning so fast. Yet it's one more example, as I've seen in our Exploration of the Brain, where there were these giants, without the modern technology, able to make amazing, groundbreaking analysis.
I like to compare him to Freud, who's another genius. But he differed from Freud in two major ways. One, he focused on only one period of life. Freud concerned himself with all aspects of the life cycle, focused only on infancy, child development. But he also differed from Freud in another way. He actually carried out experiments. This is an experiment that he did. So he really introduced experimental science into developmental psychology, which was extremely important. And he came up with some wonderful notions about this. He pointed out that not only these stages, but these stages have very characteristic features. One is they're social. That is, in order to acquire certain aspects of knowledge, and Patricia Kuhl made fantastic contributions to this, you need social interactions. So if you want to learn a language, you learn it from another human being. You cannot learn it from a television set. Moreover, these stages in cognitive development are paralleled by stages in brain development. And we can see that as kids mature, the brain grows, it adds nerve cells, it forms new connections, and these connections strengthen in certain areas, they weaken in certain areas. You can see the brain mature with time, and this parallels intellectual development.
In fact, from birth until maturity, it doubles in size.
That's right. Absolutely. And as is the case with all areas of medicine, there are tragic events that occur in a rare number of cases. Genetic mutations then interfere with these stages of development, and they can lead to Rett syndromes, Fragile X, and we'll have a chance to discuss this. So we really will see the whole range from psychology to biological basis of behavior in this discussion today. Also, one of the fascinating things that we will see is the importance of interactions between genes and environment.
For all that we will say, parents will ask, what can I do to make my child, to maximize their intelligence, their ability to learn? Can I have an impact?
Parents are born to have an impact, just as children are born to take advantage of that impact. Social interaction is absolutely critical for the intellectual development of the child. Number one. Number two, as children go through these various developmental stages, their brain is particularly susceptible to acquiring new kinds of information. So exposing children widely to numbers games, to language. If you want a child to learn a second language, it's critical that they begin this very early on. That you should be perfectly comfortable having the child be bilingual. In fact, they'll acquire a second language much, much more readily early in life than later on. And as will also emerge, enjoying the children and having the child see how pleasurable the child's activity is for the parents. So I think interaction is key for assuring a healthy outcome of it.
All right, our developing brain, that is our subject this evening. We have assembled for you a remarkable group of people, as I said, and we begin our conversation, understanding the development of the brain. Everybody, I think, understands this one big idea, that somehow it's easier for children to understand languages than adults.
Yes, that's the case. Infants go about language using both innate skills and incredibly powerful learning mechanisms that we're just beginning to understand. And whether they're born in Singapore or Paris or Seattle or New York, they'll all follow a set of universal stages, as Eric alluded to. At 3 months, they'll coo. At 7 months, they'll babble. At a year, a single word. At 18 months, 2-word combinations. And at the age of 3, full sentences, they will talk your leg off. And so what is it about that magic that they can put to work? And it exhibits what the biologists have always called a critical period in development. So if you look at your age and assess your skill in acquiring a second language, you see a very dramatic learning curve that's the reverse of what we typically expect. Adults with superior cognitive skills typically get better, surpass children at learning things. But in the area of language, from birth to 7 years of age, the kids are masters. Whether they hear a single language or 2 or 3, they will acquire them effortlessly. But beginning at the age of 7, that begins to decline. 8 to 10, 11 to 15, 17 to 39. Past puberty, which is everyone at this table,
yes.
We really don't have the same machinery. The brain is not working in the same way. And we don't learn second languages as well.
Actually, a very interesting point, because it doesn't simply apply to language.
Right.
Musical skills, athletic ability, unless you start early, you can never reach the levels that you can.
I think we're all
That's
waiting.
the worst news I've heard.
But it's awful. It's terrible.
You're a golfer.
I'm leaving.
But we're beginning to understand that many aspects of development have these critical periods. And in fact, this one, the critical period for language, has embedded in it many critical periods for the sound, learning of the sounds of the language, the words of the language, the grammar of the language.
What is it about that brain that enables it to learn language better?
Well, that's the question, Charlie. So no one debates this curve, but everyone is debating what's the cause of that function. Why does it work in that way? Why does the baby brain work in a way that the adult brain doesn't? So the classic debates, they've gone on for centuries. So currently, Noam Chomsky from MIT and B.F. Skinner, the late B.F. Skinner from Harvard, debated very different tasks, very different takes on this debate. Chomsky arguing the structure is built in the brain, and Skinner saying we're born a blank slate. So what's new, what's happened? Well, what's happened is that the scientists got into the picture and started to actually study the brains and minds of children to look at their skills. And that's produced an entirely new view that isn't either of those canonical opposites, but something much more interesting. And I can illustrate it by taking one aspect of language, the development of the sound processing of language, okay? And show you how that works. So when we think about the sounds of language, each language uses a different set. So in Japanese, R and L are not distinct. To a Japanese speaker or listener, rice and lice are the same thing.
Right.
But in English, the kids have to learn that R and L are distinct. So how in the world do they come about doing this? We have gone all over the world testing babies, and I want to show a film clip that illustrates the test. We're looking at what sound contrasts the babies at 6 months can hear in all these countries of the world. We've been in 9 countries testing with the sounds of 10 languages. And the answer is the same. The videotape will show babies sitting on a mother's lap. There's a person sitting with a box of toys and distracting the baby. The baby's got to learn that when the sound being produced in the background, ah, 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 changes to anything else, the baby's got a 3-second opportunity to turn towards the sound source and see a black box with a dancing bear or something fetching in it.
Can a baby as young as 6 months old hear the difference between 2 vowels? This baby is trained to look for the toy when the sound changes. She's being distracted, so she'll turn only when she hears the difference in sounds. The key question, will she turn before the toy lights up?
E.
So that baby knows what she's doing. She knows she's done it right. So the key answer to our tests demonstrates that the babies at birth and until 6 months are what we like to call citizens of the world. They can discriminate all the sounds of all the languages that we can put in front of them. There isn't a single one they fail at. And that contrasts with you and I and everyone here who hears the sound contrast used in their particular language but not the others. We simply can't hear those distinctions anymore. There are 2 critical pieces that babies are putting to work between 8 and 10 months. You say, what is the magic that they're putting to work that you and I can't put to work? And there are 2 very different answers. One is they're doing something we like to call computation because they are picking up statistical properties of the language that we produce for them. They're, in essence, taking into account the frequency, relative frequency of the sounds they hear. And they're also taking account of the transitional probabilities between syllables. It's an amazing computational skill. We call it statistical learning. But what we've also demonstrated is that this statistical learning capacity, which comes online at about that time, seems to be under the control of the social brain. Now, I know you've talked about the social brain on this program, so you know all about the machinery that is interested in bodily movement and eye tracking. Well, we've done experiments where we bring babies into the laboratory for the first time at 9 months, right in the critical period, for sound perception learning, and we expose them to a language they've never heard before. In this case, we're going to see a clip of exposure to Mandarin. 12 sessions between 9 months and 10 months. The babies play on the floor with my native Mandarin-speaking graduate students and postdocs. And we want to know what is it doing to their brains to get this new language. Can they take the statistics on a brand new language? And then we contrast that learning with learning that's done on a television set. Same schedule of the same dosage, same material. What happens in the learning? The dramatic finding is that only babies in the social condition learn, and they learn so well. If they're exposed to Mandarin, their sound perception becomes as good as the babies in Taiwan who've been listening for 10 months.
Wow.
So the right information in the brain at the right time in development and learning is incredible.
转1、1、2、3
Amazing.
So the babies in the television condition stare at the television set, and for all the world you think something's going on in their brains. But when you take the measures after these 12 sessions, live or television, the kids in the television condition have learned nothing.
Beyond language, what other intellectual skills?
Well, one of the things that I think is so fascinating about these years from birth to age 5 or so is that while the child is engaged in this huge task that Pat's describing of learning their language, they can't just focus on that because they also have to be learning everything else about the world. They have to go from an infant who can't even reach out and get their hand onto an object to grasp it effectively to a 5-year-old who, as you saw in one of your earlier programs, can outperform any robot in motor skills. They have to be able to come to recognize all the objects around them and figure out what they're for and what kind of actions you do with them and where you put them away, we hope they'll learn. And they also have to be able to learn basic things about how the world works, as the Piaget experiment that Eric demonstrated showed us. And I think that raises the question, what makes kids so amazing at this task? Now, the two features that Pat already focused on in the case of language, I think also apply to learning in these other domains. It's also true when children are learning about objects, when they're learning new motor skills, that they're terrifically good at focusing on other people and getting information from what other people are doing. And they're also really good at getting statistical structure, as you were saying, out of the world. But I think there's two other pieces of this puzzle, at least, that we need to add into the mix in order to account for these incredible feats that we see kids engaging in. The first of them is that children come into the world prepared to construe it in the same basic fundamental kinds of ways that we do. And that's going to be crucial, because if I'm going to learn things from you, from what you do, I better be able to understand your actions and the things that you act on in the same general kinds of ways. So let me give you just one example of this. Like Eric, I brought a demonstration in.
A learning tool.
Very simple event. I'm going to take this cracker, put it into a cup, take it out again, put it in again. You've now seen it go in and out 3 times. And over here, I'm going to take a cracker, put it into the cup, take out my empty hand, and now take a cracker and put it into the cup. Now, you saw more motion over here than you saw over here, but you'll immediately be able to say that there's one object here. Got to remind myself.
Yes, exactly.
Yes, in my prime, I can immediately say there's one over here and 2 over here. And what that ability shows is that we can take the surface appearances of things, the momentary entrance of an object, and we can understand them as manifestations of a set of bodies that exist, whether we're looking at them or not, that behave in accord with basic laws, so we can predict what's going to happen next. We know you put something in a cup, it'll stay there unless you take it out. And this general ability, Piaget called object permanence, and he rightly pointed out that this is fundamental to our understanding of the world. Well, we now know that very young infants, really as young as we can test them, within the first couple months of life, have the same ability. Now, my favorite study was actually on somewhat older infants. I think they were about 10 months old, just old enough to appreciate the virtues of crackers like this. So investigators at Johns Hopkins University presented children with these events and then asked them, what's going to be a bigger draw? Will it be the cup that had more cracker motion, more stuff that they saw, or would it be the cup with more crackers? And the babies very clearly went for the crackers, not for the cracker motion. But more sensitive methods with much younger infants, infants who don't yet know about crackers and about cups, show that already within the first months of life, babies are construing the world in terms of enduring objects, objects that they take to exist whether they can see them or not. And that ability allows infants to follow things over time, to make good predictions about what they're going to do next, so that they can begin the task of acquiring all the specific knowledge about the cups and the crackers, what's edible, what's not, what we do with different kinds of objects. So I think a piece of the puzzle of why we're such good learners is that we start from a terrific starting point, a starting point that already makes the world a coherent place for us.
What's going on inside the womb? What's going on in that brain before birth?
Before birth. So the brain starts developing quite early during pregnancy. As early as 3 weeks of the pregnancy, things will start happening. By 4 weeks, almost the first structure that will make the brain is formed. And that structure will undergo a lot of cell divisions. Many neurons will be born. They'll take their places. So that by 17 weeks of pregnancy, the brain is already starting to take the shape of the brain as we know it. And with time now, from 17 weeks on, that brain will keep on adding more and more cells. And imagine this brain being confined within a very small space, the skull. So to pack up more neurons, the brain starts contortions and gyrations will start forming, so that by the time the baby's born, it has packed about 100 billion nerve cells in that
Can you show us
structure.
a visual of that?
So if we look at the pictures, you'll see what the brain looks like at 17 weeks of age. Notice how smooth it is, that all the cells have made it in, and with time we're gradually adding more and packing more, so that by 40 weeks of pregnancy, when the baby is born, it has quite a few cells. Now this process is impacted by both external factors and internal factors. The internal factors are the genes. There are many molecules that are telling cells where to go, how to find the right place. And there are external factors, what the mother will eat during the pregnancy, what drugs she might use, what viruses she might be exposed to.
Right.
All of these factors come to play so that we have the brain as you see it. So this is what you'll see on the outside. And if you look at that gyration, that's the cerebral cortex, where motor activities are dictated, where our perception of the world around us happen. Now let's look in the inside, what happens inside the brain. And this is really beautiful because it's almost like a choreography. We start off very early on in the pregnancy where few cells are born and they have to migrate and find a place within that cortex. So the first layer of cells will be born, they'll migrate, they'll take a position. The next group of cells will divide and migrate, they'll go past the old ones and take a new position. And this process will continue till you have 6 layers. All these layers have very specialized neurons for the brain to function properly. Think of 1,000 dancers at the opening of the Olympic ceremony. Imagine them having to make the color of the Olympic flag and to have to perform a coordinated dance. If any of them takes the wrong spot, the dance will be ruined. And that's exactly the situation with the brain. We talked about intrinsic factors, genes. If we were to study animal models, we've learned quite a bit about which genes tell neurons to go to which spot within these layers. And if we look at an image from the cortex of a mouse model, you're going to notice nice layers of brain cells or neurons. They're different colors. You see the pink ones, the green ones, then the blue ones. You take away one gene, and now you can imagine how this animal might be like. The animal will have seizures, many other complications, because the brain cells have not taken their exact position. So everything has to be done precisely. Now, what happens after these brain cells take a position? They have to communicate. And brain communications continues after, the development of the pathway for cells to communicate with each other will happen after birth. So if we go back and look at the picture of the newborn baby, we're going to see the size of the newborn brain, and next to it we're going to see the size of the adult brain. You'll see how much growth has happened.
It's about twice the size?
Almost, right. And part of the reason we have such difference in size because after birth, brain development continues. It continues in many forms. First, many of these brain cells have to make different branches. They have to extend processes to communicate with each other. And if we look at the next picture, we're going to see these type of processes. The cell on the left is sending a long process, that's the axon, down which the information will be transmitted to the next neuron. The point of contact is the synapse. The synapse is where 2 neurons will communicate with each other. And that is the key factor for why we're able to learn, why we can retain part of the conversation tonight. We have to have healthy synapses. In a newborn, the synapses will start developing, but between 2 months and 2 years of age, that's when baby will have the largest number of synapses. The babies in the brain will have a lot more synapses than they will actually end up with as an adult. If you look, you'll find that the brain is actually sculpturing itself during this period. It will make so much more synapses, and with time and experience, based on the baby's experience or the child's experience, some of these synapses will be eliminated and some of them would be strengthened. And it's really tantalizing that this critical period when we see the largest number of synapses formed in the children it sort of overlaps with this critical period when language acquisition is at its best.
In fact, one way one can think about this, one doesn't really know yet, to explain the phenomenon that Pat was talking about, how one loses the distinction between certain words in a language that one doesn't use, is one loses certain connections and strengthens others.
Yes.
So the kinds of things that Huda's talking about probably participate in this phenomenon.
Yes, that's the dream, to put these two pieces of information together, the physiology, to understand how that brain is sculpting itself, and when it does that very exciting sculpting, and how that maps on the learning curves for various skills that we're very interested in, like language.
We can see not only in the language domain, but in other domains as well, two things. One is that a supportive social environment makes a big difference for children, but the other is that children have extraordinary intrinsic capacities to learn. At about the age of 2, children have two different kinds of numerical abilities that they've had since infancy. The first one I already told you about. They can represent exact numbers, small numbers exactly, the difference between 1 cookie and 2, or even 2 cookies and 3. Children at age 2 also can represent large numbers approximately. So you show them an array of 8 dots and an array of 16 dots. They can readily tell you that there are more dots in the array of 16, even when you adjust the sizes and spacing and things, so that they have to focus on number. But there are two interesting limits to young children's numerical abilities. The first is, we think of the numbers 1 and 2 and 8 and 16 as all the same kind of thing. But there's good evidence that in infants and young children, these are actually captured by separate systems that work in different ways, both in behavioral tasks and also in some tasks using functional brain imaging. They engender different patterns of activity. They seem to be separate abilities. But second, this capacity to deal with any numbers larger than 3 is radically imprecise. So a 2-year-old child can tell you that 8 is less than 16, and even that 8 is less than 12, but can't even begin to tell you that 8 is less than 9, or to think the thought, there's an exact number of things on this card. And what I'd love to do is show you a clip of a child who visited my lab 2 weeks ago, who's 2.5 years old and is just grappling with this problem of figuring out, what are the people around me talking about now when they're talking about number.
Yes.
And can you count them again for me?
1, 2, 3, 4, 5, 6, 7.
Can you put one fish in the pond?
Yep. I put 1.
Is there one fish in the pond?
Yep.
Thank you. Natalie, can you put 2 fish in the pond?
Yep.
Are there 2 fish in the pond?
Yeah.
Yeah? Thank you.
I'd like to point out she's doing 3 things that are brilliant.
Right.
Okay? The first, which is another example of learning from other people, is she's figured out a lot of what goes into this counting routine. She's got the first 3 words, at least, in order. She's pointing to objects at the right time and so forth. Second of all, she's figured out that when she's asked for 1, she should put her small exact number system to work and produce exactly 1 object. She's also learned that when she's asked for another one of those words that you use when you're counting, she should put her large approximate number system to work and produce a bunch of things. What she hasn't figured out yet is, how does she integrate what she knows about large numbers and what she knows about small numbers in a systematic, rule-bound way to yield the natural number concepts that she'll need in school and that we are using, that seem to come so naturally to us. And the fascinating story is, this is a hard task for children, but they figure it out on their own.
One of the great themes we're talking about is how to what's natural and what instinctively happened versus what is learned.
This is a beautiful example of how the two interact. Not only the social environment, but also the genes and how important they can be for the development of the brain.
Stephen, tell us.
Well, so as Huda beautifully described, the developing brain is the product of the genes. So you're born with this incredible machine that now can absorb the environment and absorb experiences and mold itself. Just as Pat said, it doesn't matter what continent you're born on, you have the ability to learn language, and it doesn't matter particularly what that language is. But we know genes play an important role because we see families that have the tragic consequence of having a particular disorder move throughout the family in a simple fashion. So we can look at family structures, pedigrees, where we represent males as squares, females as circles, and show that, for example, a phenotype, which is some recognizable characteristic or disorder, moves from parent to child and so on down through the pedigree. And that gives us a very powerful ability then to identify the genes responsible. These are rare disorders, but they give us a window into what's going on.
What triggers these genes?
Well, there's a cascade of events that happen. You know, some genes control other genes. There's a timing event during development. How cells interact when those, as Huda was describing, when the neurons move about in the developing brain, they kind of form a local environment that then communicates to their neighbors and turns on genes, turns off genes. So the repertoire of the genome is a complex orchestra of how it is turning genes on and off on cues that are sometimes environmental, sometimes driven by other genes.
What do we know about the impact of what some people call toxic elements in terms of brain development?
Environmental
Yes.
factor.
Alcoholism, that kind of thing.
Correct. So some of these things, we talked about extrinsic factors that can affect brain development. For example, alcohol can affect brain development, can cause problems with even the baby's development, defects in the midline. Some environmental toxins can affect things much earlier, actually, than just in the womb. They can affect the sperm or the egg, creating genetic defects that then will give rise to an abnormal child. So there are really a variety of factors. Viruses can cause developmental brain abnormalities. Drugs can affect it. So there are a variety of things. And part of the reason, because these elements can affect gene expression. That's how they actually work. We don't think about it, but a toxin can affect when a gene is turned on or off, either in a sperm or in utero. And that's part of the problem of these toxic elements.
So one of the really important things we need to understand is to bridge the biology that Huda and Steve is talking about to the cognitive psychology that the two of you are talking about. What kinds of headway can we begin to make in that? Is there any beginning in that?
I think there is a beginning. I think we're looking at developmental disorders in a way that says we know there's a genetic tendency, for example, in autism. We know some of the pieces that have to come together to bring about changes in these twin deficits that children with autism show in language and social cognition. So the interesting interplay in the next decade, I think, will be to understand the genetic predispositions that we're all walking around with. And then to what degree can we push them around, push around the outcomes by rearranging situations, interventions early in development when the brain is so plastic, when it's carving, its synaptic growth and pruning, and alter the time course of development in a way that would change the outcome.
And this is a question that Eric and I have had many conversations about. And how much dialogue is going on between people representing the different aspects of the psychological as well as the molecular?
I mean, both of these people are doing experiments in which using imaging or electromagnetic EEG equivalents. And perhaps you could discuss that.
So there are a couple of things to look forward to. One is that early measures, say at 6 months, we can now measure the baby's brain's ability using event-related potentials to hear the distinctions, the brain's reaction to the distinctions between sounds. That measure at 6 months of age predicts language at 3, the quality of language at 3, and reading readiness at 5. Now, that's quite extraordinary.
That's amazing.
So that says if a baby is really not hitting the milestones.
So these are like biological markers.
These are markers. These are biomarkers. So this is one thing that's happening in all of the laboratories now, where people are using the brain measures to take a precise measure of a precursor that we know is going to be very important, that this milestone be passed before you get to a more complex one. And then on the horizon are brand new metrics that allow whole brain imaging. And I've got a video of of that. Magnetoencephalography, or MEG for short, that's 23 letters, you don't have to pronounce that. MEG is whole brain imaging that's totally safe and non-invasive, can be used from newborns to aging adults. And it's picking up the magnetic field changes as the brain does its work. And so we've now been testing babies at 6 months and 12 months while they're engaged in a cognitive task. And because it's so safe, and we've got GPS tracking on their little heads so we know exactly where the brain structures are at every second in time, we can test them in cognitive tasks that involve math or learning Mandarin from a real person as opposed to a television set, and seeing how these areas of the brain communicate with one another. How does that long distance communication happen between the language areas and the social areas so that social understanding can trigger or gate the learning mechanisms for language or other tasks.
As in all areas of medicine, we learn an amazing amount from tragic cases in which the brain development is disturbed as a result of genetic disorders. And we've learned a great deal from Rett syndrome, from Fragile X, things like that. What about Rett syndrome? How does that work?
So Rett syndrome is actually quite fascinating. It's named after an Austrian pediatrician, Andreas Rett, and he discovered it because he saw these patients in his clinic, and then now we've seen many of these patients around the world. And what happens, these girls are born normal. They can learn how to do everything a 1-year-old will typically do. Some of them will actually learn how to speak few words. Some will even learn how to say nursery rhymes. They'll be able to use their hands, feed themselves, play with toys, socially interactive. And then something happens, and it happens during that critical period that we've talked about. Somehow this part of the regular postnatal developmental program gets interrupted, and these girls all of a sudden will stop using their hands, instead will just constantly wring their hands. They'll lose the social interaction, they'll lose the coordination, they will lose any language, they will lose the balance, and they'll have motor difficulty, and with time will develop spasticity, they'll have seizures, anxiety, and a variety of other medical and neurological problems. It really affects all parts of the brain. So it's quite an unusual disorder, but what it taught us that this critical postnatal developmental period is really important. And if we look at the video, we're going to show a video clip of a girl who started life as normal. You see her picking a toy, and she's picking the toy with her hands. She can run around. She's functioning just as a perfect 1-year-old would normally function. Here she is at a couple years of age going through the book. She's using her hands well. She can use the book, and she's still doing quite well. And then we're going to see her when she developed Rett syndrome. Now you'll notice she's not quite steady on her feet. And you'll see that she can't use her hands. She looks at the book, she touches it, but she can't really anymore use her hands. And instead, what she'll do with her hands is she'll hold them together and she'll constantly wring them. Doesn't have as much of social interaction. She'll have seizures and a variety of neurological problems. This happens time and time again to all these girls. And the question, what happens? And the way we were able to really start to learn a little bit what goes on in the child's brain when they have Rett syndrome is first by finding the gene. And then when you find the gene, you can genetically engineer animal models to study what happens. You can replicate the mutation that's in the human, engineer it in the mouse, and see what will the mouse reproduce the features of Rett syndrome. Having a mouse model, you can look what's happening in the brain before the symptoms develop, during, and through the course. And what was really surprising, would you think that a mouse would wring its hands if you gave it Rett syndrome mutation? And that's the case. So we're going to look at a video clip of a normal mouse, and you're going to see how a normal mouse will keep its paws apart, and then so this is a healthy, normal mouse keeping its paws apart. And then if you hold the Rett syndrome mouse model,
it's amazing.
Oh, look at that.
You will see how it will be wringing its paws together.
Wow.
And this mouse will have all the features that we see. It has learning problems, it has balance problems, it will have seizures, all the features we see in the human patients we will see in the models. So models are really very, very important. And it turns out that the protein that's lost in Rett syndrome, we call it MACP2 for short, is really critical for those synapses we talked about. So if we look at a healthy brain after all the sculpting and the pruning happens, you have to have just the right number of synapses between two communicated neurons. And this is what we would expect in a healthy brain. In the case of the Rett syndrome, what happens when the protein is lost, we have fewer synapses. So obviously having fewer synapses is not sufficient for normal brain function. We're learning more and more about one of the there are thousands of proteins at the synapse where the two neurons communicate with each other. We know that losing one copy of a gene for any one of those proteins, or having an extra copy of that same gene, can give us cognitive disabilities, autism spectrum disorders, a variety of neuropsychiatric syndromes. So the brain is really finely tuned, as you've heard from Steve, and having too little or too much is really
You see
bad.
this also in Fragile X.
Absolutely. So in the synapse, when they're forming, there are tiny machines in there that produce proteins. And that proteins are made, and this is wonderful experiments that Professor Kandel conducted, that you have to make these proteins locally, right by the synapse, in order for them to function. They will, in order to strengthen or weaken that synapse. Well, the protein that's deficient in Fragile X syndrome helps balance that. So you have signals coming into the surface of the synapse that stimulates this protein to be produced. The Fragile X protein sort of balances that out. So again, it's a situation where you need to have finely tuned amount of proteins. Too much is not good, too little is not good. And in Fragile X syndrome, when you lose this one protein as sort of the brake on the system, you now overexpress too many of these proteins. And that causes then these synapses just to be too weak.
I think this is a beautiful example of how brain sciences has come together. We're going here from a discussion of cognitive disorders in kids, normal kids. We're seeing how these various stages evolve. We're seeing how they can go awry with genetic defects or environmental defects. We can begin to study them in animal models. And one of the many functions of animal models is we can begin to define them as targets for developing drugs that reverse these. And these are terrible disorders. If we could begin to reverse them, it would have a major impact on public health policy.
So I'll show a clip of some mice that have been treated with one of these compounds. So you'll see 4 mice, and they're all engineered like Huda talked about, so they have Fragile X syndrome. If you put them in various measures of intelligence, they don't do as well as normal mice. Not that normal mice are all that smart to begin with, but they do worse. But one of the odd things that the mice do with Fragile X syndrome is they have what's called audiogenic seizures. So if you play kind of an obnoxious tone to them, they jump around all over the cage and they can't handle it. A normal mouse doesn't quite respond that way. So we're going to show a clip of 4 mice, and they're identical mice. All of them have the Fragile X mutation. But the 2 mice on the left are untreated. The 2 mice on the right are treated with one of these compounds, and then you turn the tone on, and this is what you'll see.
That's amazing.
That's amazing. It's really amazing.
So
now, it's a long way from this to a human, but this is the way you begin.
The way you begin
with having an animal model.
Oh, right.
Then you have to see safety.
Gotcha. Let me, I want to close, but I want to get to some sort of basic things here that I think everybody in our audience is curious about. They're all, Howard Gardner and others have talked about levels of intelligence and varieties of kinds of intelligence. What we know about the developing brain in terms of infants and young adults, why are there differences in terms of a numerical talent, skill, brain power, and a linguistic brain power? What causes those differences, or do we know?
Well, they're not developing. If you talk to the brain physiologists, they're not developing at the same rates. So different parts of the brain are doing their work on different timetables. And so one aspect of what's happening, that's only one, is that there really is a physiology underpinning these brain modules, if you will, that are not so modular early in development as they will become as we age. So the physiology is coming along, And then the environment, the physiology is waiting for environmental input at particular points in development. And so if that input isn't there at a particular time when the brain is expecting it, that will thwart development in these different domains.
Yeah.
And so they're not all on the same timetable.
Almost certainly the same kinds of arguments we've been hearing in other issues. That is, there are environmental factors and the genetic factors are going to be likely for these special skills. I think the most dramatic example, I'm curious to hear a discussion from the four of you, is the cognitive differences between men and women. Because certainly if you would have asked that question 50 years ago, you would have said, yes, women do this better, men do this better. I'm curious to get the modern view of this.
Let's just go around the table.
To what degree is this environmental?
Let's go around the
and
table
to what degree is this genetic?
Go ahead.
Well, I think before we ask the question, are these differences genetic or environmental, we need to ask the question, what are the cognitive differences? And this is something that I've become interested in because we do studies not only on infants and young children's understanding of number, but also objects and space and so forth. What we find in young children is enormous convergence across the two sexes. Huge similarity from one sex to another, which is not to say that everybody's good at everything or even that everybody develops skills to the same level, but the basic ways of solving problems that we see occurring in girls, we also see occurring in boys. So I think early on, a really interesting fact about human cognition is that for all of the different interests that develop between men and women, all the different physical abilities that we see on the Olympics and so forth, there's really huge convergence in the ways that we view the world and the ways that we learn about the world.
I would elaborate that also, and that is the way society treats young girls and young boys is really different. And as they become more similar, girls being exposed to wonderful competitive athletics and things like this, the apparent differences disappear. So there is, undoubtedly, there are going to be differences between men and women, but on many of these that we now see are socially determined. Wouldn't you say?
Yes, I think so. I think so. In language, we often see that girls hit the milestones earlier. But if you test them on skills that don't require speech production, so that their understanding or their perception of language, they look equivalent. And these early measures of language development that we're conducting, we see no differences between the boys and the girls.
Is there a difference in mathematical ability?
Well, let me just mention that for the disorders of language, whether that's autism or involving stuttering or other Fragile X, and some of the problems that stem from disabilities that affect language, boys are subject to the disorder at a rate of about 4 to 6 times as often as girls. So something about the brain's organization of language is going to give rise to a greater incidence in males than in females, and we don't entirely understand that.
But at a fundamental level, though, it's a difference genetically between males and females.
Sure.
So we, of course, females have 2 X chromosomes and men have 1 X. And you can do experiments with girls that have Turner syndrome, so they only have a single X chromosome, and show that they have difficulty with spatial recognition. But they perform more poorly than women do, or girls with 2 X chromosomes, but they do pretty much the same thing boys do with 1 X chromosome. So the dosage kind of will play a role.
And the parent of origin. In the case of Turner, so a girl with 1 X, her social behavior may be slightly different if that X came from the mother or that X came from the father. So there are really many layers to this genetic control, not only encoded within the sequence of the gene, but there are other variations that modify the sequence, in fact, to make it slightly different. And I get back to the really importance of the gene-environment interaction. So this critical period when a lot of brain sculpting is happening is what you're exposed to. If you're a girl and you're exposed to sports that early on, you'll probably excel in that. And if you're exposed to mathematics and reading, you'll also excel in that. So I think it's really a very strong gene-environment interaction.
A great example of that is what's called perfect pitch.
Yes.
So that's the ability.
Oh boy, you have rung my bell.
So that's the ability to just know what a musical note is by hearing it without any reference. And it turns out that's partly inherited, but it's also very dependent on early experience to formal music training. So if you have the right combination of genes and you don't have early musical training, and it's the same time period we're talking about with language, before 7 years of age, If you don't have that training, you don't have perfect pitch, despite the fact you have the right genes. If you have that early training, then you can have perfect pitch. But similarly, if you don't have the right combination of genes, but you have early musical training, you'll never have perfect pitch.
For all the either parents or potential parents watching this, what do you want them to take away from this?
They should remind themselves that they're the child's first and best teachers. And what they tend to do naturally, which is to interact socially, to speak in motherese, to play games with numbers and objects, and to allow them to explore the kitchen. You know, the pots and pans and spoons and plates are playthings that will beat the DVDs and special programs any day.
Well, maybe I'll elaborate on this and discuss, of course, prenatally, we know that many environmental factors affect the health of the brain, so be as healthy as you can be, nutrition, physically avoid drugs, alcohol, etc. But I think one thing that's really important to remember, and this is more to the parents of children with developmental disabilities, we're now learning that many of these disabilities can, at least some symptoms of them can be reversed. So the brain is really much more plastic than we thought. So I think we should hold some hope that with the research and some of these animal model studies that maybe we can do something, especially for those diseases that don't occur till 1 or 2 or 3 years, autism being one, perhaps Rett syndrome and other disorders. So I think that's another thing to be thinking about for the future.
Take the time to enjoy your child. Do it not only for your child, because I fully agree with everything that Pat said. You are the best environment for your child to be growing up in, but do it for yourself. This is an absolutely fascinating time in life, and a time when things are changing so fast that from one day to the next, from 1 week to the next, you're dealing with a different creature with new abilities. Take the time to enjoy them.
Let me then ask finally, what do you most want to know within your field? What's the biggest question you would like to see answered?
To answer that honestly, I'm going to have to turn away from questions about parents and children and say that the question I'd most like to have the answer to is, how is it possible for a human brain to think an abstract thought? You've learned on this program, you've talked about how the brain only gets input from its perceptual systems, and it only affects the world through its action systems. But there are limits to what we can see, and there are limits to what we can do. But we can close our eyes, and we can imagine a line that's infinitely long, a point that's infinitely small, a series of numbers that goes on forever. And our ability to imagine those things forms the basis of everything from formal mathematics to science to technology to everything that we are. I don't think we understand how the brain does that. I think for all the advances that have been made in genetics and neurobiology, we don't know where abstract ideas come from. But my hope is, if we could figure out how little kids get to have them, we might start making some progress.
Great.
Actually, it's very similar to you. I think that, if we could figure out how genes influence, say, schizophrenia, how do you generate a hallucination because you have a mutation in some genes? I mean, that's a profound progress from a simple gene and a protein and a synapse to a circuit that's somehow recreating an experience that was, that occurred earlier. So, I think someday to explain these psychiatric disorders that are very complex, but they have genetic underpinnings, so they must have variation in the genome. And how that occurs is really going to be interesting.
Fascinating.
I'm very interested in understanding gene-environment interactions better and co-opting that to help patients. So we know that for many of the disorders, it's really about gene-environment interaction. We're born with our genes. We can't do much about them, but we can do something about our environment. So finding ways to actually modify the function of genes through understanding these external environmental factors, and we know this could help many brain disorders, would be fascinating. Just like they're toxic chemicals, there've got to be good chemicals or good environmental factors. I'd like to know them.
I think from discussions such as this, one can see that in a reasonable period of time, we're going to get an insight into what we can do with our children in order to bring out the best in them. But in addition, we can go one step further. We've really not used science to improve the educational process. It'd be nice if we could have an impact in pedagogy on how to really optimize the experience of preschool children and children in school in order to have them assimilate knowledge better. They have this enormous capability that they're born with, and often school takes it out of them, and we need to create an environment in which kids can use this in an optimal way.
Yeah. Yes.
The brain and mind are the new frontiers. We're just on the cusp of understanding how abilities like language, which have been debated for centuries, what are the origins of language, how does that come together in an individual child? I'm looking forward to gleaning from all of the scientists that are now coming together to talk in detail about how it works on the ground, and imagining that in our lifetimes we will be able to do what all parents feel like they want to do when they look in the eyes of their child, and that is to understand what is going on up there.
A remarkable panel and a remarkable group of people. Their enthusiasm for the subject is what makes this, I think, for the audience at home and for me, their sense of the passion of learning that they experience. So next month, we'll focus on what?
The aging brain. The population is getting older, and we're going to discuss what is critical for people as they age, in addition to good health, having a good memory. So focusing on the nature of memory, how memory is stored, what are the different memory systems of the brain, how can one enhance the possibility of going into old age with a fairly good memory, and also what are the tragic consequences of having disorders of memory. We'll discuss age-related memory loss, we'll discuss Alzheimer's disease, and we'll see what we know about these disorders and how can one possibly try to prevent them in the future.
I look forward to it. Episode 6, Charlie Rose Brain Series.